Now we are right here for our Biosensors Panel. We have Professor Juan Liu, Professor Ashwin Gopinath, and Dr. Erin McConnell. So without further ado, Professor Liu, please take it away.
Okay, thank you, and welcome to the biosensor session of this conference. So we have two panelists today, and I will ask them to introduce themselves. So Erin, please go ahead.
Sure. So do you want me to just do a quick introduction, or should I share a little bit about my research as well?
I think you can just put up your slides, and then you can introduce yourself along with that.
Great. So like Dr. Liu said, my name is Erin McConnell. I'm a research associate now at the University of Ottawa. So that means I've gone through undergraduate, I've gone through all my graduate training, I did some postdoctoral research, and now I'm working more or less full-time as a research scientist. So I will show you a little bit about some of my research specialty. All right, so I've done several different kind of types of research, but they've all had one thing in common, and that is using DNA as a tool to build molecular recognition elements, meaning something that can recognize specifically a molecular target for biosensing, diagnostics, and therapeutics. And with that, I've used kind of three different types of main technology. The first main kind of technology I've used is something called aptamers. So aptamers are small single-stranded pieces of DNA. So typically we think of DNA as kind of a genetic code, something that stores molecular information and genetic information, but we can take, rather than a genome, which is billions of bases long, we can take a small piece of DNA that can be anywhere from 15 to 100 bases, and we can build these little tools out of them called aptamers that can bind and recognize really specifically a target molecule. So similar in a way to that antibodies can recognize a particular target. And so in developing these molecules called aptamers, there's this certain trajectory that we tend to follow as researchers. The first step of that is to select the molecules. So this is a process that happens in the lab. And we do it in a test tube, and we do several rounds of the same procedure. So we start out with a library of these aptamer sequences that can have 10 to the 14 to 10 to the 16 potential candidate sequences in it. We incubate that with our target of interest. So for instance, it might be a cell or a toxin or a protein. Most recently, it could be a protein that would help us identify the SARS-CoV-2 virus. So we incubate that library, we separate out the sequences that bind to the target that we want, and we get rid of the ones that don't bind. We amplify up those sequences, and then we have an enriched library that we can then use for multiple rounds of selection. And so that process continues. So once we have the sequences identified that bind well to our target, we have to understand how well. So this is the next step. This is characterization of aptamers. And so this involves really understanding how well the aptamer can bind to its target, how well it recognizes it, and how well it functions in kind of different environments. And then once we understand these things, we can apply them. And so you can apply, I like to say aptamer research goes from agriculture to zoology, kind of A to Z. And that's because there's so many different ways you can apply them. Some of what I've looked at is diagnostic tools, therapeutics, and of course, biosensors. A similar type of technology is called DNAzymes. So the type of DNAzyme that I'm interested in, and if you're familiar with Dr. Liu's research, you'll know he's also interested in these, are RNA cleaving DNAzymes. So basically, they're like an aptamer in that they bind to and recognize a specific target. But we say rather than binding, we would say that they're active in the presence of a particular target. And that's because they undergo an intramolecular cleavage event, meaning part of the sequence, the DNA sequence, gets broken off at a specific site. And you can imagine how if we have a sequence and part of it breaks off only in the presence of a specific target, for example, a bacteria, we can take advantage of that for biosensing. So same thing, we start with selection, similar process to aptamers. We characterize them. We need to understand how they work, how well they work, what conditions they work in, but also exactly what bases are making this kind of catalysis work. And then, of course, we can apply them, again, most commonly in biosensors for DNA enzymes. The last bit of research that I'm interested in, it's another type of biosensing. It's using DNA, but it's not using aptamers or DNA enzymes. It's using DNA in a bigger sense to take advantage of its programmability, its ability to form predictable structures to build what we call nanostructures. In this case, if you look kind of on the far right up at the top, you see these DB, that stands for dumbbell, these kind of shooting star structures. So what we can do is take advantage of this for sensing. So we're interested in the lab that I'm in now at the University of Ottawa, Vincent Tabarcasa's lab. We're interested in using solid state nanopores. So a nanopore is simply a nano-sized hole, so a small hole with nanometer dimensions in a thin membrane across which we can apply a voltage, and we can measure the disruption of the current. And each molecule that passes gives a kind of a characteristic fingerprint. So we can understand when DNA of a particular shape is passing through this pore. So what we can do is combine it with existing kind of sensing strategies. So that would be something similar to like an ELISA, where we're using a sandwich antibody assay. But we can take advantage of that to release a piece of DNA, which we then use to assemble a nanostructure. And if you look in the nanopore sensing in the scheme on the side here, under panel B, you see this black line with these dips coming off of it. That indicates in real time every time something's passing through the pore. What we get is this very specific signature. So when there's one piece, one P1, so a probe passing through, we get one spike. When we have the two probes connected by the piece of DNA that we recovered from the ELISA type assay, we form this nanostructure that's a dumbbell shape, and it passes through with a very different signature. So there's two peaks. And so we know from this that we can see these different types of DNA, and we can relate this to a concentration of a biomarker. In this case, we were interested in thyroid stimulating hormone, and it gives us very high sensitivity. So we were able to have a detection limit in the femtomolar range, which is super low, super exciting. So that really summarizes my research. It's a very kind of crash course in DNA, different types of DNA sensing. But yeah, that's me.
Thank you, Erin. I think that was a very concise yet comprehensive summary of DNA aptamers, DNA enzyme sensors, and yeah. So next panelist will be Professor Ashwin Gopinath from MIT. So he's an assistant professor there, and is also doing DNA-based research. So please.
Thank you for the invite. And while I just get the sharing thing get going, so much like Erin, I have gone through the whole undergrad, grad school things, and I did my undergrad in India, then was grad school at Boston University and at MIT. Then I was a research scientist, and I had a small group at Caltech for a while. Then I was at Google for a while, and then I started my lab at MIT about 2 years back. And the big question that we are interested to ask is the underlying theme of the lab is two questions. One is how finely can we control matter by self-assembly or merging that with top-down? And the second level of question is how do we actually do extremely high throughput experiments, so you can get a massive amount of data. So even if you have very weak signals, you can actually pull it out and do it in a very controllable, reproducible fashion. And the core, some of the core features of our lab is basically using this technique known as DNA origami, which takes DNA and thinks of it as a polymer, a programmable polymer to construct nanostructures out of it, onto which you can add other molecules or other inorganic nanoparticles, or pretty much anything that can be connected to DNA can be organized on this small frame. So you can think of this DNA origami as a programmable molecular breadboard on which you can start organizing, let's say, carbon nanotubes, gold nanoparticles, quantum dots, the aptamers that Erin was talking about, proteins, whatever else that you can attach it. And you can do this with extremely high yield, close to 100%. And with that in mind, our lab basically asked the question, can the DNA origami act as this bridge between top-down technologies, the technologies that is used to create all the computers, all the mobile phone semiconductor chips, and the self-assembled structures like the aptamers, proteins, quantum dots, all these other things. So can that be a bridge between the two? And as it turns out, you can actually do that. So for one of the earliest work that that I was doing at Caltech, we demonstrated that you can organize molecules at extremely high yield using top-down techniques. So the same techniques you use to create your computers, you use that, but rather than creating transistors or some other semiconductor devices, you can put a single molecule there, and you can even do chemistry onto the surface. Then we demonstrated that you can merge this with semiconductor devices. So at a given point, you can put a single molecule. And that particular picture, that Van Gogh's picture, the Starry Night, has got 65,000 different semiconductor devices. And the light coming out of any one device depends upon, is controlled by changing the number of molecules in there. And we can move from, let's say, putting just one molecule, 2 molecules, up to 7 molecules. Then we did this other experiment that we demonstrated that not only can we put molecules where we want, we can control the direction as well. This was a paper that came out last year, and wherein we can put molecules where you want and even orient it with like less than about 3 degrees error. And also, on a side note, we can also create precise nanostructures on the surface, but never doing anything that resembles nanopatterning. So completely by self-assembly, we can actually create structures with precise shapes, and it's almost any arbitrary material. And in the context of sensing, why this is very exciting is there's an entire class of experiments wherein you're doing single molecule experiments. Erin's these nanopore experiments are one instance wherein you're reading out single molecule at a time, but there is an entire other class of single molecule experiments when you put single molecules on a surface, and then you depending upon whether an interaction takes place, something else happens, it'll give out light. Now, typically, this is what an experiment looks like. You take a surface, put molecules on a surface, and then you observe what a optical signal from any given point. Now, if you increase the number of molecules, what happens is you can't be sure that the signal that you're getting is from a single molecule, because a single molecule size is going to be about a couple of nanometers, but when it's giving out light, it has about lambda by 2. If anything that is happening in this diffraction limited space, you can't distinguish it very easily. So if you increase the number of molecules, you can't get more data. If you reduce, because the quality of the data reduces. But if you reduce the number of molecules, you can be sure that it is single molecules, but you can't get as much data. So how do you optimize it? So if you want to optimize it, the only way to do it is if you can organize the molecules at specific locations and the spacing between them is lambda by 2. There are some constants lambda by 2n where there is a refractive index in there, but lambda by 2. So how do you actually organize single molecules in a hexagonal grid and guarantee that only a single molecule is there at that location? So the way in which we do that is basically using the technique that I just now said. And this is what we typically end up getting, where every single point here is a single molecule that is organized on the origami. And we know a priori that with 99.8 or some number probability that it's a single molecule from that location. So you can do more single molecule experiments than has ever been done before with the guarantee that the signal is coming. And you typically end up getting, this is just one kind of a data wherein you're observing live what is happening at each of these locations. Each of these locations, you have multiple molecules and they're moving around tethered to a particular location. So depending upon how the signal changes, you can tell certain things about the protein behaviors and things like that. That said, I'll just stop there and just, a whole bunch of people have helped me do these experiments, from Caltech to a couple of collaborations we have at UBC, ASU, and the folks who have funded us over the last many years. So I'll stop there.
Thank you, Astrid. That's a very impressive single molecule imaging, So if the audience students, right, if you have question, feel free to ask anytime, right? So I think we have got great young people here to answer your questions. Maybe I can start from the science question. So both of you talked about single molecule, right? Single nanopore, single origami, and so what do you see are the challenges to make this type of very small signal measurements, right? To go for practical application. So will they mostly just stay in the lab, or actually you see their commercialization to become device people can use?
I think we can probably both speak to different challenges. Do you want to go ahead, Dr. Gopinath?
So single molecule experiments, depending upon how one thinks about single molecule experiments, there are a lot of commercial techniques that are based on single molecule experiments. For instance, single molecule manipulation. For instance, sequencing, Illumina sequencing, even though they are not doing single molecule sequencing per se, underneath the entire process is the ability to capture and manipulate single molecule behaviors. So single molecule experiments or single molecule manipulation at scale is already sort of commercialized. And you have Oxford Nanopore and Pacific Bioscience, two other sequencing companies that are pushing the limits on what can be done on single molecule experiments, wherein you have either a single enzyme located at a particular position, and they are reading out as it's actually copying a strand of DNA. You can read out what the DNA, what the sequence is. Oxford Nanopore, they have a range of nanopores very similar to what Erin was talking about, but made out of proteins. And as the DNA is being tethered through it, you're reading it out. So for sequencing, it's already sort of well established. But single molecule sensing becomes even more important when you move into emerging omics technology, like let's say proteomics and metabolomics, wherein and the reason why single molecule becomes very important is because the concentrations of the molecules that are important, like typically, let's just look at proteomics, wherein you're trying to quantify the number of proteins in a given sample. There are about, depending upon who you ask, there's between 12,000 to 20,000 different proteins in your blood at any given time. And there are different forms. They might be in a different proteoform. Maybe they have like a disulfide bond or glycosylated or something else might have happened. So how do you actually create a table of protein and the concentrations? That requires single molecule quantification because of many reasons. One is some of the more important proteins are at very low concentrations, like femto to attomolar concentrations. So you need single molecule counting to be able to tell what it is. And also there is this subtle other feature known as dynamic range, wherein you can have molecules that are like, some molecules are at femtomolar, attomolar range, and the other molecules are in millimolar range. So you need to span about 10 to the 9, 10 to the 10 concentration in a single solution. So for that, you need to have single molecule measurements. All of these are technologies that are being pushed into commercialization, different stages. Like for instance, the work that that our lab does, that core technology is currently being transitioned through a couple of different startups for this protein counting work. So and that whole field is expected to evolve the same way genomic field emerged. So there's a real commercial potential for it, and there's a need for it. So I'll pause there, and I can speak about this forever, but yeah.
Yeah, I mean, I can add to that. So that's an excellent kind of broad picture perspective. I can add a little bit of kind of a more detailed perspective in the sense that fundamentally, at least for nanopore sensing, one of the advantages is that if you give infinite time in theory, you can detect a single molecule, a single biomarker in a sample, whereas many of the commercialized technologies that we have now are, as Dr. Gopinath said, concentration dependent. And so they need to be a certain threshold of concentration before the technology can detect it. Whereas with these single molecule approaches, given these certain set of parameters, you can, in theory, detect super low concentrations. That said, of course, there's practical limitations. It's not... It's certainly not practical, say in a clinical sense, to run something infinitely, expecting to look for something, a needle in a haystack. So we need to have a certain number of events to be able to get statistical relevancy, and make conclusions about these biomarkers. There's also some limitations of the technology in the sense that the detection range that it works well in can be limited, and so that the timing is useful. So you need to find ways sometimes to either dilute your samples or to amplify your samples so that you can detect kind of reasonably. The other advantage of single molecule sensing, and I think where one of the advantages lies here for commercialization, is this idea of kind of digital sensing. And so this idea has been adopted by a company called Quaternex. I think I always say do you know this company, Dr. Gopinath? They have basically a fluorescent platform where they look at they have a bead in a well, and there's like a single amplification event. And so Quanterix, yes, I always say it wrong. It's Quanterix. And so they've developed this massive kind of Simoa technology, they call it. And it allows for, basically it turns on. If the molecule is there, the light turns on. So you have a certain array of little wells. And if a certain number of these are turning on, it gives you an idea of whether or not the target is there. So it's not a concentration thing at this point. It's kind of a 1-0 situation. And this can allow for very sensitive detection. It can also allow for kind of multiplex detection. And that's, I think, where the power of single molecule detection lies. If we can build devices where these pores, for instance, are in parallel, we can have multiple pores in one kind of sensing device, then we can really amplify up the ability of these sensors to work. I think definitely, though, there are challenges more than I've kind of simplified things, but there are definitely challenges to commercialization. But I think that's true of any field, any sort of technology. And definitely, I think the field is moving in the right direction to kind of approach and find solutions for these challenges.
Yeah, it's a very interesting discussion, and certainly I think we see the power of nanopores and single molecule fluorescence and all these advancements. Maybe let's say a lot of the audience here are undergraduate students, right? So they may be thinking about the grad school. So what do you have any suggestion or things to tell the students, right? So how to be a successful grad student, and what graduate student life looks like?
Sure. So I guess I can start with that one. So there are kind of differing opinions or perspectives. Some people will tell you that if you want to be successful in science, you should pick a big name lab. You should make sure you go to a highly regarded institution. You should do everything you can to keep on that trajectory. My personal opinion and the other perspective is that you should first choose a mentor and choose colleagues before you necessarily choose. Obviously, you want to kind of situate yourself in the best situation as possible, but it's really important to make sure that you're going to like the people you work with, because that can make you more successful than having a bad relationship in a really kind of well-known or prestigious lab or location. And so what I always say is, shoot for the stars, but make sure in the process that you're also reaching out to students who are already in the lab, or you're reaching out to kind of new graduates from that lab, just so you can get a good perspective. You should talk to the professors who run the lab. You should talk to, if they have research staff, you should talk to them. If they have students, you can talk to them, and oftentimes the contact information is posted on the website. So you can freely kind of reach out to people. You can talk to people at conferences. That's a great way to kind of interact and network and get an idea of whether or not your kind of personal learning style or learning preferences and research preferences align well. You just want to make sure you have a good fit and you don't singularly consider kind of the prestige of the research. That's what I would say.
Sort of good advice.
And just to add to what Erin was saying, I mean, so I can't stress it enough that don't go after brand names. They help, but if you are in a really large lab, it might sound good, but the reality is that you will get to meet professors probably once a semester, maybe even less.
If you're lucky.
If you're in a 40, if you're lucky, if you're in a 40, 80 people lab, you will never get it, but you will have resources. And so that's one thing. And the other stuff is, I can't stress this enough. It took me a long time to kind of get this through, wherein like try to be comfortable enough and sort of to have your perspective on the research and look for problems rather than techniques. Like for instance, if you are interested in, right now CRISPR-Cas9 is all the vogue, so everybody runs into labs that work there. But that's a technology that doesn't necessarily mean that you are working on a problem that would excite you. There might be so think of defining the problem that you want to work on, like early on, rather than looking for a professor who has name and who's working on the hottest thing right now. Look for a problem and be comfortable enough to kind of try to define that problem. Oftentimes, if you define the problem very precisely, the solution kind of pops out automatically, and then you know what you want to do. Because it's 5 years, if you're not excited by it, it becomes really, painful. So
yeah. It's a really long 5 years.
Yeah.
Absolutely. I agree with that. Yeah, wholeheartedly.
Yeah, 4 years in Canada, but still, right? So that's base time in your life. You want to, to use it wisely. There's a technical question here. Is it practical to use nanopores for whole genome sequencing? I think people are doing that, right? That Oxford nanopore. So sometimes the problem is not how long you can sequence. It's actually for the sample preparation, right? To actually maintain the integrity of DNA, like millions of base, right? So they may get just cleaved at some random places, right? So then but the nanopore technology can indeed read very long DNA strands.
Yeah.
Another question: what is some foundational advice you would give a student wishing to break into the field as they think about their future career? So that's not necessarily grad school. So maybe there are other paths.
So yeah, I mean, I think the best way to I'm assuming here when you say break into the field, I guess you're meaning biosensing in some sense, or basically, so if you're thinking about biosensing, if there are depending upon there are tons of companies that are trying to do biosensing at different levels of sensitivity, plexity, and all of that. But I guess the best way to start in any of this is basically if you have background in anything related to biology, anything related to like nano sciences, nanofabrication, or anything in optics or electronics, it doesn't, I mean, the thing is, it is so interdisciplinary. You need to have multiple areas. Like you need to be comfortable enough to move from one area to another. So what is so it doesn't matter whether you're in biology or some engineering or what part of engineering. You can actually move in here because it brings in perspective from your particular background, I guess. And that's true for most companies entertain and encourage students with varied backgrounds. So just find a company or a professor or an area that you find interesting, and then just basically jump in it. I mean, jump headlong into it and see what happens, because you will learn there isn't any course right now that can give you that full landscape of everything that can happen in biosensing. It is very interdisciplinary. So regardless of where you're coming from, you just need to be comfortable enough to kind of pick new skills.
Yeah, absolutely. I would support that. Even in my own experience, my undergraduate degree is in neuroscience. My PhD is in chemistry. My first postdoc was in biochemistry and biomedical sciences department. And my research associate position now is working in biophysics. So the thing about kind of moving around, getting different perspectives, is it's like, people are diverse. People bring diverse ideas, but so do these kind of disciplines. And so if you can be trained to look at a problem from a discipline specific perspective, and as many of those perspectives as you can bring together, it can help you come up with unique ways to approach these challenges in biosensing.
Yeah, I think that kind of also partially answered another question. So what sparked your interests in these branches of nanotechnology? So I think everyone is different, right? So they will have their own, university they enter, and then there they will see different professors, right? So that's when you go to a university, right? So then there are a few options, and I personally think curiosity is the most important element. You want to be curious. You want to be actually really interested in the topics, right? So, and you get those interests, and one is based on probably your own experience, and then you have to dig deep into the project, and don't be afraid of failure. So I think a grad school is a great place to fail, and the cost of failure is fairly low, right? So you can get trained to handle problems, and so when you start to be able to solve problems, then I think you'll feel more confident and more interested in what you're doing.
Yeah, absolutely. And that's so important. The one thing you're guaranteed in science is that you will fail. So you need to be able to kind of learn from each of those experiences and be able to kind of think, okay, I planned this experiment. Everything I know tells me it should have worked. Why didn't it work? So you're going to learn something new. And so absolutely, I think what Dr. Liu says about not being afraid to fail is probably in my mind, the best advice you can get going into biosensors, but really any field in science, especially.
Yeah, and just to add to that, I mean, I think so. So in my case, when I came to the US, I started working in neurobiology with a specific question, then got tired because I didn't realize I'd never planned for basically dissecting mice. And somehow that I didn't take that into consideration at all. So I started my PhD and then basically flipped to applied physics, and that's when then there were a lot of experiments that I did. And while I was going through the experiment, I realized that I could never do single molecule experiments the way in which I wanted. I could never actually put molecules where I wanted and create a experiment that I actually trusted, wherein I can be sure that things are happening the way in which I want. So it was a problem. that basically took me to change field to bioengineering, to go and go pick up a whole bunch of skills as far as DNA and nanotechnology is concerned to solve this problem. I still haven't solved that problem. In the pursuit of trying to actually solve that issue, you develop tools, and while you're going through that, you realize that there are other problems that you can apply your technology for, and you just basically stumble your way through it. So which is why I didn't realize it at that point. What is more important than technology, a particular technology or a particular field, is having spent time thinking about a problem that you want to actually contribute to solving. It doesn't have to be single molecule biosensing or biosensors. It could be a whole lot of other things, and it allows you to get comfortable with flexibility, because sometimes if you want to solve a problem, you will have to go and learn things in a completely different field. It's just the way it is. So don't try to limit yourself to, let's say, 2D materials or graphene or DNA sensing or nanopores or something. Like, just be one level up, because it gives you a lot of flexibility.
Yeah. There's also, I would say, you learn the more and more experience you gain in science is that there's limitations to funding also. So say you want to solve a problem of a really rare disease, but maybe it has similar mechanisms to cancer. So you do cancer research, you learn these skills, and then when you become an independent researcher, you can apply them to whatever you're passionate about. That said, I don't think I know a single researcher who started out in one field and has continued literally linearly in that singular focus all the way into their career at present. So I think it's definitely, there's lots of opportunities to kind of shift gears, and you definitely need to be able to kind of go with the flow and follow kind of the science and see where it takes you.
Another question popped up. What do you all find most rewarding about your work in this area?
Okay,
Erin,
so
you want to start?
Okay, there you go.
Yeah, sure. Okay. So for me, I'm really interested in applications in healthcare. And so I started out in research because I was interested in neurodegenerative disease, because I had a personal connection to it. I wanted to understand the mechanisms of how it worked. I wanted to be able to build therapeutics, better diagnostics to understand. And so what I find most rewarding about the field of DNA nanotechnology in particular is the immense promise that it has for versatility in answering all of those questions. I find that there's DNA tools that can kind of be used for all of those different problems to help us understand lots of different things in medical care. And so I find that really rewarding, but I also just really love doing science. I love being at a bench. I love having a hypothesis and testing it, coming up with answers, seeing kind of new things. Like sometimes when you get really specialized, you're the first person who's ever seen or understand this sort of experiment that you're doing. And so I find that just really fun, that I get to do what I find fun every day in the lab, and I get to learn new things every day, and I get to meet new people. I get to hear new expertise. Like even just watching Dr. Gopinath's presentation, I thought, I do some DNA origami, but I want to make Darth Vader. Like that's so fun. So just to be able to do things like that, I find you can have a big picture reward, but it's important to find happiness and kind of positive emotions in kind of the small things as well.
Yeah, I mean, for me, it is very similar, or it used to be for at least about 4 or 5 years back. Basically, I was playing with the single molecule stuff because I'm an optical engineer by training. optical physicist by training, and it was all interested in trying to do single molecule microscopy and single molecule fundamental physics with that, because that's why many of those papers are around trying to do quantum information processing and something like that. And about 4 or 5 years back, there was like a personal event wherein I had to go through a whole bunch of different health issues, and it became very, very, very clear to me that that there is really no technology that allows you to actually tell somebody's health state at a given point. That is, and it's also very true from an asymptomatic, all the things that you have seen around COVID stuff, and it became clear to me that sequencing doesn't actually tell you the information. Somebody's DNA doesn't tell you anything about a person's health at that particular point. A good example of it is if you get a chest pain and you go to an ER, knowing your sequence doesn't, your genome doesn't give you any information. But if I could actually know the concentrations of every single protein that is there in your body, and if I knew all the small molecules that are concentration of small molecules that are there, that has a direct implication on what is your health state. It doesn't tell you just about one particular organ system. It can literally tell you everything about an individual's health. So then the question becomes, how do you actually build technologies or tools that allow you to kind of do that? And that's where it comes, you need to develop, like being able to actually do single molecule experiments at scale and at cost becomes very, very important. And pretty much all my lab effort basically has pivoted to that, solving that problem over the last 4 or 5 years. So the question is, if I take a drop of blood, can I actually count all the proteins and all the small molecules in there? And possible, and do that all in, like, let's say a couple of hours. And then it becomes how do you interpret it and all of that stuff. So what is more rewarding? I'm solving that problem. And whatever it takes to actually solve that problem, that's the mission. So, yeah.
And that's what gets you excited. And to me, I think just small things, right? Get me excited. Like what I developed, what I discovered, like Arian said, is used by other people, and they tell me in person, right? When we bump into the conference, your method worked, right? So helped my research, that really made me happy. So, final question: what's some fields of research that are adjacent to yours in biosensing? So I think that has been mentioned a couple of times, right? So biosensor is really a big field, and you have the chemistry, biochemistry, engineering. So a lot of them are needed to put together a biosensor. I'm not sure if you have anything to add.
Yeah, I was just going to say something similar in that I kind of presented a really simplistic view of aptamers and DNA enzymes, especially as a technology. And even within that field, there's people doing so many different things. So like I did my PhD at Carleton with Dr. Maria DeRosa, who does kind of agriculture and therapy type applications. I know Dr. Liu is doing kind of environmental stuff with aptamers and DNA enzymes. Yingfu Li at McMaster University is doing DNA enzymes for healthcare and environmental monitoring as well. So there's so many kind of adjacent fields in my specialty. It's hard for me to really kind of summarize some that are even outside of that. I would say in my case, in my interests, it would be just oligonucleotide therapeutics. So like ASOs, mRNAs, silencing RNAs, these sorts of ideas. They're more application based though than biosensing, but they need to be detected for a lot of disease states.
I mean, I just it's similar to what Professor Liu kind of pointed. Pretty much any area other than probably high energy physics and probably condensate physics or like condensed matter physics is basically adjacent to biosensing, because pretty much every other field you can take one step and be applicable to biosensing. So
Yeah, it's a very broad field. Okay, so I think that's all the time we have. Thank you both, and thank also the students who, I guess, participated and raised the questions. So I think our session is now ended.
Yeah, thank you so much, Professor Liu, Professor McConnell, and Professor Gopinath for being here. We all learned so much and really, really enjoyed your panel. So thank you again for being here for the conference. For attendees of the conference, we're now moving on to our breakout sessions with Andrew Barretton and Seamus Bannon in the sessions tab, same as last time. So Andrew Barretton is a senior computational scientist. He's working at Cyclica Toronto Inc. and has his PhD in biophysics. He's super passionate about modeling and machine learning, and he's going to talk about that and its applications in nanotechnology in his breakout. And then Seamus Bannon is a battery engineer at Cuberg in San Francisco. He is actually a nanotechnology engineering alumni from the University of Waterloo, and his batteries are based on an innovative non-flammable electrolyte combined with a lightweight lithium metal anode. So two really exciting breakout speakers coming up. So please attend those and head over to the sessions panel. And also, please don't forget to check out the posters and vote if you haven't already. Once again, thank you so much to our panelists. We really enjoyed your panel. That was such a wonderful experience. We learned so much. So thank you guys so much.
Great. Thanks for having us.
Yep. Bye.
Bye.